Hey guys, so I know that the amino acid metabolism whole thing is really complicated and there can be a little bit confusing with all of the different amino acids and every single one of theirs pathway and trying to keep it all straight. I remember I had a little difficulty with it, but I came up with this mnemonic back in biochemistry that kind of helped me keep all of them straight and keep them all like confined in one single mnemonic. So I just wanted to share it with you all. It might be helpful for some of you who are struggling with it. So it goes like this. Thanks, Lucy. I very much have protein too. So the thanks, Lucy, the first letters of each one of those words stands for the essential amino acids. Those are the ones that you basically cannot make or synthesize. You have to take from your environment. You have to get it from food. And so those are tryptophan, leucine, isoleucine, valine, methionine, histidine, phenylalanine, and threonine. And so thanks, Lucy, I very much have protein too. Also, the order that I have them in helps me group them into different groups. So for instance, the ones that I have in yellow right here, those are all the glucogenic amino acids. So those are the ones that can be used to go and make glucose. Whereas the ones in green, leucine, technically it's leucine and lysine. So this L stands for leucine and lysine. Here, these are the two ketogenic amino acids. And finally, I've got another grouping here of the L, I, and V, the leucine, the isoleucine, and the valine. Those are in blue are the ones that are used in the branched chain alpha ketoacid dehydrogenase to go back into the TCA. So let me just start. So once you have those all written down, you know them that way, and you have the different groupings because they do test on the ketogenic and the glucogenic, and this is also very high yield for STEP. Everything I have on this slide is very high yield for STEP as well as your biochemistry class. So leucine, going through this dehydrogenase, the branched chain alpha keto dehydrogenase, can be turned to acetyl-CoA. Isoleucine and valine together instead get turned into propanil-CoA. Now this is where it's important. Acetyl-CoA can go into the TCA cycle to get ATP. Well, it's going to be turned into NAD, then it goes through the electron transport chain to make ATP. However, propanil-CoA over here has to go through a few steps before it can go into the TCA. And so propanil-CoA goes through a carboxylase using biotin. Okay, this brings up a good point that I wanted to mention as well. So for biotin, which is B7, B6, and tetrahydrobiopterin, which is BH4, those three, a good way, just as a good rule for you to keep all of these straight and it'll help you remember things, is biotin goes with carboxylases most of the time. B6s go with decarboxylases most of the time. And BH4 usually goes with hydroxylases, again, most of the time. These are not strict rules, but the majority of the enzymes that they help out with or go along with are usually are those ones. So by B7 carboxylases, B6 decarboxylases, and BH4 hydroxylases. So here propanil-CoA will go through propanil carboxylase using biotin to be turned into methylmalonyl-CoA. Then it'll go through a mutase which uses B12. So B12 is used into two big enzymes, and here's one of them. So you should remember this one and that one. Those two are pretty high yield, this whole little mechanism that's going on here, this pathway. So propanil-CoA to methylmalonyl-CoA to succinyl-CoA, and then succinyl-CoA goes to succinyl dehydrogenase, which is part of the TCA, to go on and make ATP through the electron transport chain. So those are your branched alpha keto acid dehydrogenase pathway. If this is messed up, you end up getting maple syrup urine disease. And maple syrup urine disease will always be presented on a test with a kid whose pee smells like burning sugar. That's how it's always presented. So that's maple syrup urine disease. Now let's turn our attention to the M. So we've covered L, I, and V. So M, methionine. So if we go over to the side over here. So I have my methionine pathway. This is a very important pathway. So methionine is turned into S-adenosyl methionine, which is turned into S-adenosyl homocysteine, which is then turned into homocysteine. Homocysteine is an important molecule. not because it does anything for us. It gets used to make methionine again in this pathway. However, if we end up having a breakdown of this pathway and we build up homocysteine, homocysteine is highly thrombogenic. It's a procoagulant. It will cause a person to just throw thrombi left and right. It's hypercoagulable state if you have homocysteinuria. So that's why it's dangerous and it's not good to have. So homocysteine, so you have two things going on here. Methionine is going down this way and making homocysteine, and over here you've got THF, which is tetrahydrofolate, which is coming in from folate. So we take in folic acid, we can't make it. Bacteria can make it, but we can't. And tetrahydrofolic acid will go through multiple steps and will make N10 formyl THF and then go down to make N5N10 methylene THF, which is going to turn into N5 methyl THF. You don't have to memorize this whole pathway. But what I want you to remember here is that N10 formyl THF, this guy gets used in the purine synthesis. And I'll explain why that's important in just a second. Now, this is all the purine synthesis is part of your biochemistry, so you should definitely review that pathway. But remember that N10 formyl THF gets used in that pathway. So down here, once N10 formyl THF turns into N5 methyl THF, it mixes with homocysteine, and together they go through methionine synthase using B12 again. So here's the second enzyme where B12 is important. Remember I mentioned the mutase up here. Now here is the other one, B12 with methionine synthase. Together they make methionine and THF again, and this just kind of replenishes the cycle. And the cycle just keeps going this way. And part of the time we'll make N10, and this will get used into the other pathway. And this way, like I'm using this for this pathway, and also homocysteine can be turned into cysteine down here. So that's what I'm using this whole cycle for, is to kind of fuel my purine synthesis and making cysteine. But let's say I can end up breaking down this pathway in two different ways here. I can either have a defect in my methionine synthase, or I can have a B12 deficiency. If I have an issue here, not only am I going to build homocysteine, but I'm going to be making N5-methyl THF. I'm turning all of my THF into N5-methyl THF, which is not being used because this is broken down. So if my methionine synthase isn't working, I can't mix these two together. So I've got things stuck as homocysteine, I've got methionine being stuck as homocysteine, and I've got THF stuck as N5-methyl THF right here. So what I end up getting is homocysteinuria, but at the same time also get megaloblastic anemia. So I think there's a big misunderstanding, and it's easy to confuse things here between pernicious anemia, B12 deficiency, and megaloblastic anemia. All three sometimes are used interchangeably, but they're all separate things. So when we say megaloblastic anemia is a case where your cells, your homopoietic cells, especially your red blood cells, are not dividing all the way down. So as you start off with a really big cell called a blast, and it tends to be very big, and it breaks down and it divides, and as it divides down, it makes smaller and smaller and smaller cells. That's how homopoiesis works. But if I don't have enough DNA components to make all my divisions, I end up stopping too early. So I end up having a blast that's not fully developed, so it's a blast, and it's a megalo because it's big, so it's a big blast. And so that's what megaloblastic anemia is basically red blood cells that have not divided all the way down, and it's because I don't go through all the divisions because I don't have enough DNA. And that's because I don't have N10 formyl THF, because I'm getting it all stuck down here as N5-methyl THF. So that's what megaloblastic anemia is. Pernicious anemia is where your body creates an autoimmune reaction that attacks your parietal cells in the stomach, and the parietal cells no longer can produce that intrinsic factor which protects B12 and allows it to be absorbed in the ileum. So if you're not eating B12, we call that a B12 deficiency, and that usually occurs with vegetarians. And keep in mind, you would have to not be eating B12 for several years, almost 10 years. Your body's very good at storing B12. So that's a deficiency. B12 deficiency is when you're not eating B12. Pernicious anemia is where your body's not making that intrinsic factor because of an autoimmune condition. And megaloblastic anemia is the final result of not having B12 and not having enough N10 formyl THF to make purine synthesis right here. So those are the three different terms: megaloblastic, pernicious, and B12 deficiency. I hope that clears that up. But so again, the idea here is if I don't have B12, I'm going to end up getting homocysteinuria and megaloblastic anemia. If I have MET synthase deficiency, again, I'm going to get megaloblastic anemia and homocystinuria. Another way that I can get homocystinuria is if I block down this, if I don't have enough B6. So right here, there's an enzyme, cystathionine synthase, which uses B6 to make cystathionine and go down this pathway. So that's the other way I'm getting rid of homocysteine. If this breaks down, I can build up homocysteine and end up with homocystinuria again. And again, it's not good because it causes thrombosis, and you don't want that. Okay? I know that's a lot, but just go through it a few times. Finally, so then the next one is the H. So that's histidine, and the only thing that you need to know here is that, again, histidine, B6 with a decarboxylase, like we said as a rule, histidine becomes histamine. And histamine is important because it helps with your allergy reaction from your mast cells. Okay, next up is tryptophan. So tryptophan, again, another one of the essentials, and it's important because tryptophan, and here look again, also another rule to kind of help you remember these pathways. If I'm starting out with an amino acid, especially when I'm talking about tryptophan and when I'm talking about phenylalanine, usually you start off with an amino acid, and if you want to turn an amino acid into something else, you always go through a hydroxylase and BH4 to make that something else. And that something else, for it to turn into a third molecule, use a decarboxylase and a B6. And this pattern is seen with these two especially, with tryptophan and phenylalanine. And I'll show you. I know it's confusing when I'm just saying it. But so tryptophan right here, for me to take it down to 5-HT, which is 5-hydroxytryptophan, I'm using hydroxylase and BH4. And then to go from the 5-hydroxytryptophan to serotonin, I'm using a decarboxylase and B6. And then serotonin can be turned into melatonin. So that's why tryptophan is important, because it makes serotonin one of my important neurotransmitters. And then, of course, melatonin, which helps regulate your sleep cycle. And that's made in the pineal gland. Tryptophan is also used, and it uses B2 and B6. You don't have to know the enzymes. Just know that tryptophan helps you make niacin. Why is this important is because we have a disease called Hartnups, and that's basically where your gut does not absorb neutral, nonpolar amino acids like tryptophan. And so if you can't absorb tryptophan very well, then you're not going to be making as much niacin. So you're going to end up getting pellagra, which you should know about pellagra already. So dementia, diarrhea, death, and dermatitis. And the dermatitis that presents usually is around the neck. It's called like a collar, and it looks like a sunburn and also feels like a sunburn. So that's pellagra. Death, dermatitis, dementia, diarrhea, dermatitis, and then finally death is the fourth thing. So that's it for tryptophan. Finally, phenylalanine. Phenylalanine does a lot. So the first thing that you have to know with phenylalanine is, again, look, an amino acid being turned into something else, the first step is always a hydroxylase with a BH4. So phenylalanine to tyrosine, use that hydroxylase, phenylalanine hydroxylase. And if that's deficient, you get PKU, phenylketonuria. And phenylketonuria is very important because it can cause severe mental retardation if it's not discovered early on. And actually, we do a screen for all newborns to check on PKU. So PKU, usually the way it's presented on a test is it's going to be a kid and they happen to change the diaper and it'll smell like a mousy odor. That's how usually they'll present that, or a musty odor. And so phenylalanine to tyrosine. Tyrosine is a non-essential amino acid, but if I have phenylketonuria, the way I'm going to treat that is I'm going to basically cut phenylalanine out. And if I cut phenylalanine out, then I've got nothing to make tyrosine from, so then tyrosine becomes an essential, only if I've got PKU. But then tyrosine can turn into multiple different things. So the first thing that it can go down, the first pathway, is it can go down and make homogentisate. And homogentisate goes down. Homogentisate actually goes and turns and goes and joins the TCA. But there is an enzyme down there, the one that before you get to homogentisate is homogentisate deoxygenase. And if I've got a deficiency there, I get alkaptonuria. And notice I made it in black because you end up having cartilage that's black, and your urine will also turn black after a few hours. Now, it's not really like a terrible disease because people tend to have normal lives, but they will have some joint issues, and some people also develop some painful issues as well, like just joint pains and things like that. But it's not a massively terrible disease. But the big thing is that they'll present you as either they'll show you cartilage that's black or they'll tell you the urine turns black. The other thing that tyrosine is important for is it makes T3 and T4. So it goes on to make the T3 and T4 that's on thyroglobulin. So that's your thyroid hormone, and that's very, very critical for your body just in general. The other thing, and they love to test about this just on STEP and even on biochemistry, and even it'll come up in histology, but tyrosine goes through an enzyme called tyrosinase, which is very aptly named, to make melanin, and that's how we get albinism. So if that's deficient, then you can get albinism. Anyway, back to the other pathway. So tyrosine, again, another amino acid. For it to be turned into a different molecule, you go through a hydroxylase and BH4. And then DOPA goes through a decarboxylase and B6. So remember how I said an amino acid, if an amino acid is turning into something else, you have to use a hydroxylase. And then that next thing for it to turn into something else usually is a decarboxylase with B6. So that's the case here. And DOPA goes to dopamine. Dopamine is turned into norepinephrine. Norepinephrine turns into epinephrine. And all three of those guys are extremely essential for your body. And you'll learn more about that as you continue your medical school career. The last thing I want to mention here, and this I don't think it's big for this test particularly, but norepinephrine to turn into epinephrine uses an enzyme called PNMT, and that uses SAM. So SAM is basically this little molecule that helps transfer a methyl group. So that's all it's used for, and it's used in multiple areas. But this is one of them, and it uses PNMT. And this enzyme is really important because it's in your adrenal medulla. And so epinephrine is adrenaline, and that's the main place where that's made. And so PNMT is found in the adrenal medulla. I just want to mention that because it is a high yield point that does come up later on. It'll come up in physiology, and it'll come up also in STEP. But yeah, so that's it. I hope that helps. I hope I didn't take too long. But yeah, good luck, guys. And also I have like a key right here of everything that I just said. So just remember, B6 is usually a decarboxylase, B7 is usually a carboxylase, and BH4 is usually a hydroxylase. And everything else is kind of explained here. And I'll post all of this. All right, thanks, guys.